Good morning, students. So today we shall going to start a very important topic of central nervous system, that is opioid analgesics. You can expect a lots of theory as well as viva question from this top chapter. And to start with opioid analgesics, there are some terminologies. We should be have clear concept about that. Opioid, opium, and opiate. What are they? So opium is the plant. Actually, it's a word derived from the opus. It's Greek word for juice. So opium is a plant from which opiate is derived. And the term opiate refers to the compounds structurally related to the products found in opium. So opiate refers to all the compounds which are structurally related to products found in opium. So opium has multiple products we can obtain from that, and opiate refers to the compounds structurally related to them. So natural opiates can be natural or it can be prepared synthetically. So natural opiates are derived from the resin of the opium poppy. The scientific name is Papaver somniferum. And opiates include the natural plant alkaloids. The examples are morphine, codeine, thebaine. So these are the naturally obtained opioid. Morphine, codeine, and thebaine. Papaver somniferum is the source, and we can have other semi-synthetic products which are structurally similar to this morphine, codeine, and thebaine. An opioid is any agent, regardless of the structure, that has functional and pharmacological properties of an opiate. So opioid can be any agent which act on the opioid receptor, and it can have structural similarity with opiate, may not have structural similarity with opiate. So, but pharmacological properties and functional properties are same as an opiate. So, in short, all opiates are opioids because opiates are the products which are obtained or can be synthetically produced and have some structural similarity with opiate. So opiates are structurally similar, also functionally similar, and they are obtained from the or maybe semi-synthetically prepared from outside, and they act on the opioid receptor. And opioid is any agent which, regardless of the structure, but has functional property, pharmacological properties of an opiate. So all opiates are opioids, but all opioids are not opiates. Understand? Because all opioids are not opiates, because some opioids may have structural similarity, may not have structural similarity, but they are functionally similar. So all opioids are opiates. All opioids are not opiates, but all opiates are opioids. That means opioids are a broader term. It can have structural similarities like opiate, it may not have structural similarity like of opiate, but functionally they are same. And opium is the plant, and all opiates are obtained from the opiates are either obtained from the plant source or can have structural similarity like an opiate. And opiate and opioid both act on the opioid receptor. So make it clear, opiates are obtained from the plants, may be synthetically prepared, and they are structurally same like the drugs which are obtained from the plant. And opioids can have structural similarity, may not have structural similarity, but functionally they are similar. So opioids can be any agent, may not have structural similarity. Okay, so once again, all opiates are opioids, because opioids are broader term, but all opioid are not opiate. Okay, the opioids which have structural similarity like an opiate or products obtained from the opium, they are called only opiates. So opium is the plant, opiate is the products obtained from the plant source, and opioid is any agent which can have structural and functional similar, which may not have structural similarity, but functionally they are same like opiate. Okay, so I'm not going into detail with history, you can find it in slides. So you just remember that morphine, the most commonly studied opioids, it is named after Morpheus. He's a Greek god of dreams. Okay, so there are three receptors are there: mu, kappa, and delta. And opioids, previously it was thought that it is obtained from the plants only, but this Cosilides and associates identified an endogenous opiate-like factor that called enkephalin. So in brain also we have some endogenous opioid. They are called enkephalin, endorphin and dynorphin. So these are called endogenous opioids, these three agents, enkephalin, endorphin and dynorphin, and they act on the different opioid receptors. So students, what are the opioid receptors? Mu, kappa and delta. These are the three opioid receptors present in our body, and we have endogenous opioid also. These are the enkephalin, endorphin and dynorphin. Now opium derivatives. What are the opium derivatives? So opium is a dark brown. What is opium? Once again, opium is a plant, and actually opium is a dark brown resinous material obtained from the poppy capsule. So poppy capsule is the poppy's plant. The scientific name is Papaver somniferum. In this poppy plant there is a capsule, poppy capsule, and opium is a dark brown resinous material obtained from the poppy plant capsule. So this is the opium, and it contains two types of alkaloids. One is phenanthrene derivatives, one is benzoisoquinoline derivatives. Phenanthrene derivatives are morphine, codeine and thibane. Morphine quantity is maximum. This percentage can be asked in Viva. Morphine 10% present in the opium, codeine 0.5% and thibane 0.2%. Thibane is non-analgesic, morphine, codeine are analgesic. And we have benzoisoquinoline derivatives like papaverine, which is non-analgesic, and noscapine. So these are the actually natural opiates which are obtained from the opium. Okay. So this is the source. This is the, in left side you see the poppy plant, and in right side this is the poppy seed, and from the capsule there is a resinous material obtained which is called opium, and within from the opium we have some natural opiates. This is the morphine, codeine, thibane, and all the opiates acts on once again the opioid receptors, but opioids are the broader term. They have structural similarity. They have functional similarity with opiates, but may not have the structural similarity. So these are the mu, kappa and delta receptors. Radioligand binding studies have divided the opioid receptor into three types, and each has a specific pharmacological profile and pattern of anatomical distribution in the brain, spinal cord and peripheral tissue. All of them are G-protein coupled receptor rhodopsin family. So what is the net effect of agonist binding on the opioid receptors? The mu kappa delta receptor largely couples to the pertussis toxin sensitive GIGO proteins, but occasionally maybe GS or GQ. And upon receptor activation, the GIGO couplings results in following effects: inhibition of the adrenal cyclase activity, reduced opening of the voltage gated calcium channels, stimulation of the potassium current, and activation of the protein kinase C and phospholipase C. So basically opioid drugs, they tend to reduce the transmission of the pain signals. So once again they are inhibitory to some of the neurons. So how they exert this inhibition? One is by inhibition of the adrenal cyclase activity, reduced opening of the voltage gated calcium channels, stimulation of the potassium current, and activation of the PKC and PLC. So these are the actions of different types of opioid receptors, mu, kappa and delta. Mu is predominant. Mu is predominantly responsible for the analgesia, supraspinal and spinal. Supraspinal means action which is exerted above the spinal cord level. That means at the brain, and spinal means which is exerted at the spinal cord level. Respiratory depression is another effect. Sedation, euphoria. It is specifically obtained by mu receptor. That means the pleasurable activity effect, which is the reason of actually the drug addiction. Meiosis, reduced geomotility. Kappa on the other side, C, it causes dysphoria and some psychomimetic effect. Also it can cause sedation, respiratory depression. On the other hand, delta, this is responsible mainly for this affective behavior and reinforcing action, for which this is also responsible for the drug addiction and abuse. So mu receptor, where they are present, this is major receptor mediating the actions of the morphine. These high densities of mu receptor is obtained in the is found in the periaqueductal gray thalamus, nucleus tracta solitarius, nucleus ambiguous, and area postrema. Right? And it has a specific inhibitor antagonist, that is a beta phenyltrexamine. And mu receptor once again is subdivided into two varieties. Mu1, it is responsible for the supraspinal analgesia, and mu2, it has a lower affinity for morphine, mediates the spinal analgesia. Once again, supraspinal analgesia means analgesia mechanism which is active in the level above the spinal cord, and spinal analgesia means action at the level of the spinal cord. Okay, so mu receptor present in this following location: periaqueductal gray, thalamus, nucleus tractus solitarius, nucleus ambiguous, and area postrema. Kappa receptor. So mu receptor, remember mu receptor, the endogenous opioid for mu receptor on which mu receptor act is called endorphin. Now kappa receptor, it has a high affinity for dynorphin, another endogenous opioid. So mu receptor acts on the endogenous ligand is endorphin. Kappa receptor endogenous ligand is dynorphin, and it has a specific antagonist, norbinyl torphamine. It has two receptor subtypes, kappa 1 and kappa 3. Delta receptor, its endogenous ligand is enkephalin. It can be leucine or methionine, leu-met enkephalin. And the delta mediated analgesia is predominantly spinal. An affective component of supraspinal analgesia appears to involve delta receptors. Affective component is mainly psychological component, and it's present in the limbic areas responsible for the dependence and reinforcing action. So in the limbic area of our CNS, delta receptors are predominantly present, and it is responsible for the dependence and reinforcing action for which patient is may be indulged into repeated intake of these drugs. Myenteric plexus neurons express high density of delta receptor, mediate reduced GI motility, and it has a specific antagonist called naltrexone. So what is the pharmacological actions of the morphine? It is a site-specific depressant and stimulate action on the morphine in the CNS by interacting primarily with the mu opioid receptor as a full agonist. So before starting, so analgesia is its predominant effect. So to understand how it causes analgesia, we have to study in short about the pain pathway. So morphine is a strong analgesic. Dull, poorly localized visceral pain is relieved better than the sharply defined somatic pain. And nociceptive pain arising from the stimulation of the peripheral pain receptor is relieved better. Apprehension autonomic effects are dampened, and suppression of the pain perception is selective without producing proportional generalized CNS depression. So dull, poorly localized visceral pain is relieved better than the sharply defined somatic pain. So sharply defined somatic pain, they are better managed by the NSAID. So mechanism of pain. So under normal conditions, pain is carried by the small diameter C and A delta fibers. Unmyelinated C fibers are unmyelinated, they convey a dull diffuse burning pain, and A delta fibers are myelinated, they convey a sensation of the sharp well localized pain. And C and A delta fibers convey nociceptive information from the muscle viscera as well as from the skin. So whenever there is an injury to the muscle or viscera as well as skin, C and A delta fiber carry the pain to the higher centers. So modulation of the nociceptive pathway, look here. Peripherally there are multiple factors like heat, cold, pressure, tissue injury, which stimulates the afferent pathway, and bradykinin, serotonin, prostaglandin, ATP all stimulate the afferent transmission of the pain in the afferent pathway. Substance P and CGRP, these are the another few mediators, and all of them are transmitted to the dorsal root ganglia. The cell body of these afferent pain fibers present in the dorsal root ganglia. From there it goes into this dorsal horn. So this is the dorsal horn of the spinal cord, and from there they cross the site and go to the contralateral spinothalamic tract. Okay, so this is the peripheral pain mechanism. Bradykinin 5-HT, prostaglandin, ATP, and also CGRP, substance P, they carry the pain transmission, pain from the afferent pathway to the dorsal root ganglia cell body. From dorsal root ganglia they go to the dorsal horn, and then to contralateral spinothalamic tract into the thalamus. So the cell bodies of the spinal nociceptive afferent fibers lie in the dorsal root ganglia, and C and A delta fibers enter the spinal cord via the dorsal roots ending in the gray matter of the dorsal horn. So dorsal root ganglia contains the cell body of the C and delta fibers, and there they are then these are projected into the gray matter of the dorsal horn, mostly in the superficial region, which is called substantia gelatinosa. And other A fibers penetrate deeper. There are multiple 6 lamina in the dorsal horn. So A and C delta fibers mostly projected into the lamina 1 to 4 and in the substantia gelatinosa, and it is rich in both endogenous opioid peptides. After that, nociceptive afferent neurons release glutamate, ATP, substance P, and CGRP at their central synapse in the dorsal horn. So these are the mediators which can be inhibited to inhibit the pain transmission. And after they are reaching the afferent fibers reach the dorsal horn, ascending nerve axons travel in the contralateral spinothalamic tract, as has been shown by the previous picture, and they synapse on the neurons in the ventral and medial parts of the thalamus. So from the dorsal root ganglia, they go to the dorsal horn, then via the contralateral spinothalamic tract, they reach the thalamus, ventral and medial part of the thalamus, from which they are further projection to the somatosensory cortex. So this is the ascending pathway of the brain, and if we can inhibit any of the route, we can inhibit the pain transmission or sensation of the pain. There is also a descending pathway, and it starts from the midbrain, brainstem, inhibitory effect on the dorsal horn transmission. So remember, descending pathway inhibit the pain transmission via the ascending pathway. So if we want to decrease the pain sensation, either we have to inhibit the ascending pathway or we can stimulate the descending pathway, because descending pathway sends inhibitory effect on the dorsal horn transmission. And electrical stimulation of the midbrain periaqueductal gray area causes analgesia. So midbrain contains a specific area that is called periaqueductal gray. Stimulation of this area sends inhibitory signals to the ascending pain transmission pathway. So descending inhibition is mediated by the endogenous opioid peptides. Once again, there is a role of the 5-HT, serotonin, noradrenaline, and adenosine. And opioids cause analgesia by activating this descending pathway or inhibiting the transmission in the dorsal horn, that is the ascending pathway, or inhibiting the excitation of the sensory nerve terminals in the periphery. So this is the descending pain control mechanism. Periaqueductal gray present in the midbrain. It is the major center which sends signals to the ascending pain pathway, and this is the periaqueductal gray receives the input from the higher centers, and it is also the main output center of the limbic system. It projects to the rostral ventrolateral medulla, and from the RVM, that is the medulla, descending inhibitory fibers project to the dorsal horn of the spinal cord. So once again, so if you remember, the dorsal horn is the pathway where all the ascending pain fibers are projected from via the dorsal root ganglia. So here also in descending pathway, from the periaqueductal gray, signals are sent to medulla, that is the rostroventral medulla, and from the medulla it goes into the dorsal horn of the spinal cord. And if you stimulate the periaqueductal gray, then dorsal horn is stimulated, but this descending pathway stimulation actually inhibits the signal transmission via the ascending pathway. That means from the dorsal horn, then there will be less projection to the spinothalamic tract, less projection to the somatosensory cortex, so there is less pain sensation. So what is the supraspinal mechanism of analgesia? That means which is active on or exerted on the level above the spinal cord. Mu-opioid receptor agonists block release of the inhibitory transmitter GABA from tonically active PAG. So GABA, periaqueductal gray are tonically active that regulate projection into the medulla. PAG projections to the medulla activate medulla-spinal release of the NEN5-HT at the level of the spinal dorsal horn. So periaqueductal gray, that is present in the midbrain, they are normally active and they are normally releasing GABA. Okay, but mu-opioid receptor block release of the GABA from the PAG. So what is the effect? So GABA is inhibitory. If inhibitory neurotransmitter release is inhibited, then periaqueductal gray is stimulated. So normally periaqueductal gray is under GABAergic control, that means it is inhibitory. but MAOP receptor is inhibiting the GABA release. That's thereby it it stimulates the periaqueductal gray, and periaqueductal gray, if it is stimulated, they sends more projections to the medulla, and if in the medulla there is a release of Ne and 5-HT, which is sent to the spinal cord dorsal horn. So that is the medulla spinal pathway. So PAG projections to the medulla activate the medulla spinal release of the Ne and 5-HT at the level of spinal dorsal horn, which attenuate the dorsal horn excitability. So mu receptor mainly stimulates the descending pathway, and descending pathway stimulation actually helps in preventing transmission of the ascending pathway. So how they inhibit? So periaqueductal gray is normally under GABAergic control. They inhibit that MAOP receptor, and thus there will be less GABA release from the periaqueductal gray. So actually periaqueductal gray, there will be more projections of the the periaqueductal gray projections to the medulla activate, so there will be more stimulation of the periaqueductal gray, which activate the medulla release of the Ne and 5-HT, and this excess Ne and 5-HT are reached to the spinal cord dorsal horn level, and this release can attenuate the dorsal horn excitability. So this is the excited at the level above the spinal cord. Now what is the spinal action? Spinal action of the opioids, they selectively depress the discharge of the spinal dorsal horn neurons evoked by the small efferent nerve fibers. Majority of opioid receptors are associated with small peptidergic primary afferent C fibers, and remainder also present in the dorsal horn neurons. So spinal opiates reduce the release of the primary afferent peptide transmitters such as substance P contained in the small afferents. So spinal action of the opiates, actually they reduce the release of the primary afferent peptide transmitters present in the afferents, substance P. So this is actually easy to understand. So they release the primary afferent peptide transmitters like substance P, also CGRP contained in small afferents. And presynaptic action prevents the opening of the voltage sensitive calcium channels, prevents transmitter release, and postsynaptic action blocks excitation of the dorsal horn neurons directly evoked by the glutamate. So in spinal action, we can divide it into two parts, presynaptic and postsynaptic. Presynaptic is easy to understand. They prevent the opening of the voltage sensitive calcium channels, so prevents transmitter release. Transmitter here is substance P or CGRP. And postsynaptic action is blocks the excitation of the dorsal horn neurons directly evoked by the glutamate. And there is the activation of the potassium channels in the postsynaptic neurons, hyperpolarization, and this joint capacity account for the powerful and selective afferent of opiates in spinal nociceptive pain processing. So in short, mechanism of analgesia by morphine, it can be spinal and supraspinal. Spinal action, mainly there is decreased release of the substance PCGRP by presynaptic action, decreased opening of the calcium current, and postsynaptically it causes hyperpolarization and it causes decreased stimulation of the glutamate pathway, and supraspinal level it causes stimulation of the periaqueductal gray by inhibiting the GABA release, thereby it increases the medullary release of the Ne and 5-HT, which are sent to the spinal cord dorsal horn, and by this mechanism there is decreased transmission of the ascending pathway from the dorsal horn to the contralateral spinothalamic tract and the subsequent pathway. So if ascending pathway is blocked, then pain transmission can be inhibited. So morphine acts by this mechanism. Okay. There is a peripheral action, but not clinically significant. Direct application of the opiates to a peripheral nerve can cause local anesthetic-like action at high concentration. This is not naloxone reversible. In the absence of inflammation, there is no peripheral effect, and it is mediated by opiate receptors on the peripheral terminals of the small primary afferents. Local opiates in the knee joint and in the skin can reduce the firing of the spontaneously active afferents. So, question till now, what you should answer it. All opiates are opioids, but all opioids are not opiates. Is it true or false? All opiates are opioids, but all opioids are not opiates. It is true or false? Number 2, what are the receptors of morphine? which receptor is responsible for the euphoria and which receptor is responsible for the dysphoria. Number 3. So which receptor is responsible for the euphoria, which receptor is responsible for the dysphoria. Number 4 question. What is the main area of the CNS where morphine causes analgesia by morphine causes supraspinal analgesia. So supraspinal analgesia, which part of the brain is mostly responsible? This is the number 4 question. And number 5 question, which is, what is the scientific name of the poppy plant? What is the scientific name of the poppy plant? So we shall continue. So after analgesia, there is another effects of the morphine, that is called sedation. So sedation is different from that produced by the hypnotics. It can cause drowsiness, indifference to surroundings, and higher dose progressively induce sleep and coma. So that is normally not obtained with the analgesic dose. If we are increasing the dose, then there is sedation. Another is inhibition of the respiratory center. So morphine depresses the respiratory center in a dose dependent manner. Rate and tidal volume both are decreased, and initially you remember that there is a depression of the ventilatory response to the increased CO2. So initially when there is increased PCO2, there is less stimulation of the respiratory center. So normally excess PCO2 stimulates our respiratory center, but in high dose of morphine, this stimulation of the respiratory center to higher PCO2 is depressed. So it is mediated by the depression of the excitability or mainstream chemosensory neurons. Later hypoxia drives to the respiratory center are also suppressed. So initially there is less stimulation to the hypercarbia. Later hypoxia drives to the respiratory center is also suppressed. So hypoxic stimulation of the chemoreceptor is effective after the decrease in the responsiveness of the CO2. So when there is less respiratory stimulation with excess CO2, if we give if oxygen supplement, then hypoxia drive is also abolished. So that can cause apnea. So we should be careful about that. So excess CO2 is the reason of respiratory depression. We should not correct it with by hypoxygen, okay? Because hypoxic drive is actually stimulating the respiratory center till then, but inhalation of oxygen may remove this residual drive and produce apnea. And there can be increased gestural rigidity. If you remember the general anesthetic class, fentanyl, that is an opioid group of drug which is used for the IV general anesthesia, that can also cause gestural rigidity. And morphine in higher dose also cause increased gestural rigidity, causing further difficulty in the respiration. There is diminished upper airway patency because of the morphine is one drug which releases the histamine, so there is bronchoconstriction. So morphine causes decrease in the respiratory depression by these three mechanisms. One is by decreasing the respiratory center, mainly decreasing the sensitivity of respiratory center to the excess CO2. Then it causes increased gestural rigidity, and also it causes bronchoconstriction. So there is diminished upper airway patency. Now there is a common question: what morphine stimulates some center in the CNS and some depresses other centers. So what are the other center depression apart from respiratory center? So apart from respiratory center, it depresses the cuff center, it depresses the temperature regulating center. Hypothermia occurs in the cold surrounding, and it depresses the vasomotor center, for which it causes fall in the BP. Also there is little bit decrease in the heart rate. So cuff center, temperature regulatory center, vasomotor center, and respiratory center. Remember this. So respiratory center, cuff center, temperature regulatory center, and vasomotor center. These four centers are inhibited by the or depressed by the morphine. Okay? And stimulation of the CNS center by the morphine. One is CTZ. So morphine, opiate or other opiates sensitize the CTZ to vestibular and other impulse. It causes vomiting. Edinger-Westphal nucleus, that is the nucleus of third cranial nerve. It is stimulated by the morphine. It causes meiosis, and morphine overdose can cause significant meiosis. That is also called pinpoint pupil. So no meiosis occurs on the topical application of the morphine to the eye. So because the meiosis is a central effect, it cannot be obtained by the topical application of the morphine. Vagal center is stimulated. It causes bradycardia, and certain cortical areas and hippocampal cells are stimulated, convulsion may occur. So these are the 4 centers by which is stimulated by morphine. Most important are CTZ, it causes vomiting, Edinger-Westphal nucleus, it causes meiosis, and vagal center stimulation, it causes bradycardia. Okay, and certain cortical centers are stimulated which can cause convulsion. So once again, recapitulation: morphine depresses which center? Respiratory center, vasomotor center, temperature regulating center, and cuff centers. That's why morphine can be used as antitussive agent. And morphine is stimulated which center? CTZ, Edinger-Westphal, and vagal center. It can cause mood alteration and rewarding properties. It can cause euphoria, and behavioral pharmacological data point to a pivotal role of the mesocorticolimbic dopamine system. So how this rewarding mechanism acts? So mesolimbic dopamine system originate in the ventral tegmental area and projects to the nucleus accumbens in the forebrain. So remember these two area, ventral tegmental area, and which is projected to the nucleus accumbens, and there is dopamine projections from the VTA and glutamate projections from the prefrontal cortex. So if you see the picture, it will be clear. So this is the rewarding mechanism. So this is the PFC is prefrontal cortex. It is projected to the nucleus accumbens, NAC, and from NAC it is projected to the ventral pallidum, and this is the ventral tegmental area. So PFC releases glutamate, VTA releases dopamine, and NAC is under GABAergic control, and all of this NAC is projected to the ventral pallidum, and whenever there is excess discharge from the ventral pallidum, there is excess reward. That means patient is having some reinforcing effect, patient is have some euphoria, which is driving patient to take further drugs which may not be medically appropriate, and this is the reason for addiction. So how it acts? So look here, mu opioid receptor present in here, here, and here, and so what is the main purpose? Main purpose is to stimulate this ventral pallidum. Now this is GABA. So if GABA is inhibited, then there will be more discharge from the ventral pallidum, more reward. So mu opioid receptor inhibit this nucleus accumbens. So the GABA release is inhibited. Mu opioid receptor present in the cell body as well as in the presynaptic area, and there is decreased release of the GABA. And also they are influencing the dopamine release also. Now here glutamate, we know glutamate is excitatory. So glutamate prefrontal cortex stimulation means more glutamate discharge, and more glutamate discharge here, nucleus accumbens is releasing more GABA. More GABA release is we do not want, because more GABA release will actually decrease the discharge from the ventral pallidum. So glutamate here is excitatory, but dopamine is inhibitory. So mu receptor causes stimulation of the dopamine. So ventral tegmental area is stimulated by the mu receptor. Excess dopamine is actually inhibitory to the GABA. So when GABA is inhibited, then discharge from the nucleus accumbens is inhibited, so there will be more discharge from the ventral pallidum and there will be reward phenomenon. So remember here, dopamine is inhibitory and glutamate is excitatory, we all know, GABA is inhibitory. So when inhibitory is there, negative is positive, so there will be more discharge from the ventral pallidum. So what is the function of the mu opioid receptor activation? Mu opioid receptor activation causes decreased GABA release from the nucleus accumbens, and there is increased dopamine release from the ventral tegmental area, VTA. So increased dopamine release actually causing decreased stimulation of the nucleus accumbens. So far there will be decreased GABA release, and actually there will be more stimulation of the ventral pallidum and more reward phenomenon will happen. So this is explained in this slide. Dopamine delivered directly into the nucleus accumbens is powerfully reinforcing. Opiates increase the dopamine release in the nucleus accumbens. So opiates increase the dopamine release, which is actually inhibitory to the nucleus accumbens. In the nucleus accumbens, mu opioid receptors present prosynaptically on the GABAergic neurons, and this reinforcing effect is caused by inhibition of the local GABAergic neuronal activity, which otherwise acts to inhibit DA flow outflow. So remember this pathway: prefrontal cortex glutamate is stimulatory, VTA dopamine this is inhibitory, and mu opioid receptor increase the dopamine effect and inhibit this nucleus accumbens, So there is decreased GABA release from the nucleus accumbens and ventral pallidum is stimulated, causing excess reward and reinforcing effect. So this is the rewarding mechanism or reinforcing effect, and this is exhibited in the mesolimbic dopamine system, and main pathways are ventral tegmental area and the nucleus accumbens. Okay, so I shall finish here, and the questions are number 1 or number 6. Actually there are 5 previous questions. Number 6 question: what is the effect of mu opioid receptor on the nucleus accumbens, whether it is stimulated or whether it stimulates nucleus accumbens or inhibit the nucleus accumbens. Number 7, what is the center stimulated, cuff center, it is stimulated or depressed by the morphine. Number 7, cuff center is stimulated or depressed by the morphine. Number 8, vagal center, whether it is stimulated or depressed by the morphine. Answer these questions and we shall move to the next session.